All right. So this is the last of the fungal lectures, and then we're going to move into plants next week. We have finished grading the tests. The grades will be posted sometime soon. They still haven't been posted, and you can get them back next week. Don't get used to it. The mean and median on this test is very high, relatively speaking. Not sure if that's just because you're all brilliant or exactly what, but I warn you, the next midterm will be harder. We're doing, there's going to be lots of detail for all these life cycles to learn with fungi and a lot more detail in plants. So we're going to be, this is to warm you up into the course and to get all the background needed for the rest of the course too. But hopefully, even though the next midterm might be a little bit harder, the mean will be the same. But we'll have to wait and see. And I'll post, just for if people are curious, I'll post this as the histogram of the distribution of scores. And then, you may not know this, but we get back for the Scantrons, not just the average for each question, but how the top 25% in the class did on those questions and how the bottom 25% did. And that sort of tells us about the difficulty levels of the questions. And I'll also post this too. There were 2 questions that a lot of people did not do quite so well on, but all the other questions, the average was reasonably high, and the top 25% did very well on. So we're going to move into the last discussion of fungi now. And after telling you at least a little bit about fungi that kill things and fungal pathogens, what we're going to talk about today is fungi that have either beneficial effects in mutualistic associations, or fungi that humans use in some way, or if we get to it at the end, fungal effects on the global ecosystems. There's a little movie trailer, I'll post the link to this too, on mushrooms saving the planet, which is basically what we're going to talk about. I'm going to go through we'll probably get through the first 4 of these topics today. What I really want to focus on are these mutualistic associations. That's the primary point of today's lecture, is different examples of mutualistic associations involving fungi. And then we'll talk a little bit about some of these other topics for other positive impacts of fungi on humans and things humans care about. So you should remember mutualisms, symbioses where both of the partners benefit. And one thing that's sort of really useful to think about when we talk about mutualistic associations, if you have some idea that there is a mutualistic association between 2 organisms, you don't always know what the partners are providing for each other. And it can be really helpful to understand the biology of the individual species as well as the biology of whole ecosystems to try and figure out what benefits the 2 or more partners in a symbiosis are getting from each other. And that's what I'm going to try and highlight with the examples we're going to talk about with symbioses involving fungi. So one of them I already started on, but I'm just going to go through again, and I have a video to show that has a little more detail on lichens. So lichens are symbiosis involving fungi and a photosynthetic partner. That photosynthetic partner is sometimes an algae in the Plantae group. We will talk more about those algae in the next few classes. And sometimes the photosynthetic partner is a bacterium, usually a cyanobacterium. And there are also other microbes that are sometimes involved in lichen symbioses that are not photosynthetic. But the key mutualism is between a fungus and photoautotrophic other microbe. And one of the really interesting things about lichens is by having this symbiosis where one of the partners is really good at acquiring water and nutrients, that's the fungus with hyphae that have large surface areas, and a photosynthetic partner that can fix carbon, and frequently also those organisms can fix nitrogen, the photosynthetic partner in some of these symbioses, and these allow them to colonize incredibly harsh environments, including, for example, just surfaces of rocks where you wouldn't expect organisms to be able to grow particularly well. In most of the lichen symbiosis, basically the way it works is there's fungal hyphae that provide a top and bottom layer, and then intermediate layer where the photosynthetic species is sort of housed and kept and protected, and the fungus again brings in nutrients and water, and the fungus and the photosynthetic member fixes carbon and makes sugars and other compounds. And I thought I found this really good video from Science Friday where they went out and did a little field project interviewing someone about lichens and telling a little bit of background about lichen biology. And again, I will post links to all of these if you want to.
The leaves are already falling off the trees, and winter's just around the corner. But that's no reason to stay indoors. Science Friday is taking you on a field trip. We're headed to the Tannersville Cranberry Bog in Tannersville, Pennsylvania, to learn all about lichens. Our tour guide today is James Lindemer. He's a lichenologist at the New York Botanical Garden. He comes to the bog to collect his specimens.
It's sort of like a puzzle, finding them everywhere you go, and learning what they look like. I think of myself as a bounty hunter.
If you'd like to hunt for this bounty yourself, you probably don't have too far to look.
Lichens can be found in every terrestrial habitat on virtually every substrate that's available. They can grow on trees, they can grow on moss, they can grow on soil, they can grow on rocks, and new substrates that are introduced, like human-made substrates like plastic or cars or glass. If they sit still long enough and they're in the right conditions, lichens will grow on them too. I described a new species, Gyrolebiopsis woodii, the tire rut in southern New Jersey.
So the biggest problem with looking for lichens isn't finding them. It's knowing what's a lichen and what isn't. Lichens fall into three distinct groups. There are the foliose lichens,
They look like a leaf.
the fruticose lichens,
It looks like a little bush.
and the crustose lichens.
Those are the ones that pretty much look like a crust on a tree or a branch or a rock.
And they come in lots of colors. But beware of false alarms.
Here's an example of a species that's not a lichen, but is treated with lichens because it belongs to a group that contains lichens. And that's Penicillium polyporium. Look at that painting. Yeah, pretty.
So what exactly is a lichen? It turns out that lichens aren't just one thing. They're actually groups of three different organisms: fungi, algae, and bacteria.
Basically, these three components get together and form a constituent organism that doesn't look anything like any one of the individual parts. The sort of master of the whole thing is the fungus. The fungus is really the farmer in the lichen.
The algae are essentially the crop that's being harvested. The fungus eats the algae, which produces energy from sunlight. In return, it provides it a home, protecting it from excess UV radiation. While scientists aren't certain exactly what the bacteria do, it's known that all these organisms work together.
What lichens really are sort of a term for a lifestyle.
And many species have adopted that lifestyle.
In any given one place, you can have 50, 60, 100 species.
But despite the fact that there can be so many varieties and are found practically everywhere. Scientists are only just beginning to understand them.
Lichens represent something that you can see in your backyard that are very poorly known.
Take this common yellow lichen, called noon glow. It's found throughout eastern North America, but it was only described by scientists last year.
Someone probably would have collected this, but, it's just a thin film, and most lichenologists might have thought it was an alga, might have ignored it because they thought it was an alga, and most people that work on algae would probably have ignored it because they said, oh, it's a lichen.
Which means that if you want to become a lichen hunter, there's a whole world out there for you to discover. For Science Friday, I'm Rothschild, and I'm Lichen.
Haha. All right, because he's just like a bounty hunter. And I mentioned before, there's lots of environments where people have come up with various uses for, just going to turn down the volume here. Various uses for lichens, including as dyes. I must have taken out the slides. People eat lichens too. Actually, I'll show that when I talk about food in a little bit. So that's just one example of a symbiotic partnership involving fungi. And there's another example, which we've already talked about briefly when I talked about the different groups of fungi, which are Mycorrhizae. And the mutualism here works in conceptually a very similar way to what lichens are doing. There's a photosynthetic partner, in this case, plants, and there's a fungal partner, and the fungus does basically some of the same general things it does in the lichen symbiosis, helps bring in water and nutrients, and the plant provides food. And there are many, so mycorrhizae is basically fungal root associations. There are actually quite a diverse array of different kinds of mycorrhizal associations, and I'm going to just tell you briefly about a few of those. So there's one called Ectomycorrhizae. Ecto means on the outside, and this is a case where the fungus basically wraps around, the hyphae wrap around roots, and then they reach out into the surrounding soil, and that basically serves as a way of increasing surface area for the plant root system. But they don't actually go into the plant cell walls themselves. They're basically just wrapping around the outside of the roots. And there are been a few of these Ectomycorrhizae symbioses that have been characterized. There actually aren't that many that are known, but there are a few hundred that are out there that have been characterized. And they've been found involving both Basidiomycota and Ascomycota. And again, there's a reasonable number of different species of fungi that have been found to be involved in this. If you look across all plants, it's about, I'm not sure of the exact numbers, but something like 3 to 5% of plants that have been looked at carefully have been found to have some sort of major ectomycorrhizal symbiosis. And examples include oaks, the Quercus genus, Betula, which are birch, eucalyptus trees, and there are a few others. And it's in these cases, these ectomycorrhizal symbioses are critical for the survival of these particular trees or plant species. And then a second type of mycorrhizal association is one I've already mentioned, because there's a whole lineage of fungi that all of the members of this group are involved in this symbiosis, and this is the Arbuscular mycorrhizae. And they are the ones I mentioned before that actually can get into the cell wall of a plant. They don't go into the plasma membrane, but they get inside the cell wall and then wrap around the plasma membrane. And so it's a very close association between the fungus and the plants. And again, the main purpose here appears to be the plant providing sugars and various nutrients that are made from the energy that the plant gets via light, and then the fungus aiding in acquisition of water and nutrients from the soil. I was looking for a video, the equivalent of the fecal transplant, but I couldn't find one. But if you're doing planting of plants, and there's lots of agriculture where people try to basically add, either directly or indirectly, both the ectomycorrhizal species or the arbuscular mycorrhizal species, and this can greatly aid in the growth of individual plant species. Some of them basically are nearly dead without them. Some of them will survive, but their yield will be incredibly low. And then, so again, there are lots of these associations where the mutualism appears to be driven by the surface area that fungal hyphae can create and the ability for them to bring in nutrients and water from the soil and the plants to feed them. But there are many other mutualistic associations that have been found involving a diversity of fungi. I'm just going to show you one of those here that is particularly cool and particularly interesting. But again, there are hundreds upon hundreds of different mutualistic associations between plants and animals, and even between fungi and some bacteria and microbial eukaryotes. So the one I'm going to show you about it, we also have a little video. It's Friday. I figured a few videos wouldn't kill me or you. So this is a really interesting interaction where ants farm fungi.
Leafcutter ants make their nests in underground chambers. They emerge regularly to forage, blazing trails that extend hundreds of feet into the forest. Most tropical plants are permeated with toxic chemicals, a deterrent against browsers. The ants cut fresh vegetation, but they don't eat it. They feed it to another organism. Foragers carry their cargo down into the nest and turn it over to smaller worker ants. They clean the leaf fragments and chew them into a pulpy mulch. Leafcutters cultivate a fungus that breaks down the toxins in the leaves and swells with proteins and sugars. This is the ant's food.
Both the ants and the cultivated fungus are dependent on each other for living. The ants need the fungus as a food. They're dependent on it, and take away the fungus, they will die. In reverse, the fungus cannot do without the ants. So it's a mutual codependency.
A mature colony of leafcutter ants can consist of as many as 8 million individuals, and they're the dominant herbivores of the New World tropics. They take an estimated 15 to 20% of all the fresh vegetation. A mature colony of Atta leafcutter ants are the equivalent of an adult cow sitting in the middle of the rainforest, foraging on the vegetation in their immediate area.
The entire rainforest is affected by the symbiosis of ant and fungus. To understand how it evolved, Schultz and Mueller collect ant nests throughout Latin America.
Here's one.
An experienced eye can spot the subtle signs of a young nest, founded perhaps 6 months ago when a new queen left home with a bit of fungus in her mouth and burrowed into the ground.
There it is.
Opening a nest is a very exciting moment. Suddenly the cavity opens and you see the fungus fighting. And then you may see the queen.
There's a queen here. Dude. The size of a peanut.
What we've learned from studying the ants is that you can have a long-term resistance over 50 million years as an agriculturist. There's clear parallels between the ant agriculture and human agriculture. Both types of societies are dependent on cultivation of some other organism and have very sophisticated procedures how to promote the growth of these organisms.
But human farmers are plagued by pests in their crops, while the ants' gardens seemed pest-free. A century of research had concluded that the ants are probably so adept at weeding that no infestation can take hold. A graduate student in 1998, Cameron Curry, just didn't buy it.
I actually actually had some people tell me that looking at diseases in the ant gardens was kind of a silly project, that the ants maintain their gardens free of diseases, and so why would you be going there to look for diseases? So I went out and collected ant colonies, and I isolated pieces of the garden to see what was there other than the fungus the ants cultivated.
He cultured 1,500 fungus samples, and the same aggressive mold kept showing up. When he removed the ants from the nest, he saw the mold devastate the fungus in a matter of days. So the ants did have a pest in their gardens. But how did they keep it so completely under control? Cameron began to wonder about a white, waxy coating on the body parts of some ants. What really intrigued him were the ants working deep in the gardens that were covered with the stuff. He asked the experts about it.
In the past, people had just considered this to be some sort of nondescript secretion that was produced by the ants for unknown, probably uninteresting reasons.
And Cameron was the first to put this waxy secretion under the microscope and noticed it was not inert, lifeless, but it was actually alive.
The wax turned out to be tangled mats of bacteria. But what shocked Cameron was that these were the same types of bacteria that produce half the antibiotics used in human medicine.
I remember my graduate advisor and I were laughing, thinking that wouldn't this be exciting if these ants have been effectively using these bacteria for production of antibiotics for millions of years, when humans only discovered this 60 years ago, and we thought at the time that this was maybe a bit far-fetched.
Far-fetched, but true. It seems the ants have been using antibiotics to control the pest in their gardens for some 50 million years. So why hasn't the mold evolved antibiotic resistance?
I think that the answer probably lies in the fact that the ants are using cultures of millions of cells of bacteria to produce these antibiotics. And so these bacteria are evolving. Likewise, the pathogen that is the target of these antibiotics is also evolving. And it's an evolutionary arms race that has continued for 50 million years.
And so the symbiosis of ant and fungus also includes the aggressive mold in the fungus garden and the bacteria living on the ants. Nature is often more complex than it first appears.
Scientists have just begun to understand how two species can interact, 3, 4.
Sorry, no Ed E.O. Wilson here. So anyway, this is a pretty amazing sort of example of agricultural-like systems in animals, and the farming that these ants do is actually very detailed and elaborate in terms of how they move the fungus around, how the queen, for example, they just briefly mentioned this, the queen has to carry a little bit of fungus with her to the next colony, just like brewers carry a little bit of yeast or bakers carry a little bit of sourdough starter culture. That's exactly what the queen ants are doing when they go start a new colony. And so there's now some really interesting work going on trying to characterize in detail all the different microbes that are in this partnership. And so, for example, if you look at the phylogeny of the ants, at least in this paper from a few years ago, the ants that do some type of farming are all a single monophyletic group. So there was some origin of this behavior to develop the agriculture-like system, I can't remember how many millions of years ago, and then all the descendants of that sort of original evolutionary event have continued to do some type of fungal farming, although the exact system that they do is different in these different clades. So it's a really fascinating example, and although we're not going to go into it, there are lots of other examples of systems where it appears that insects and sometimes other organisms like snails are involved in some type of leveraging the biochemical properties of fungi in order to digest cellulose. So animals aren't able to digest cellulose on their own, and we need other organisms to help us do that. And so there are a lot of different ways to do that. Some termites have microbes in their gut that do this for them, and others appear to not only have microbes in their gut, but also have some that they keep on the outside that help them digest plant material. And the example, they didn't mention this in detail, the bacteria that this guy Cameron Curry first characterized are in the Actinomycete group, the high GC gram-positives, and they produce a diversity of antibiotics, antifungal compounds actually, that are used to kill these molds, but don't kill the fungus that the ants are farming. All right, so are there any questions about these mutualisms before we move on to other topics? All right. I just thought I'd give a brief mention. We talked a little bit about studies of the human microbiome before, and most of what I showed you, most of the work that has been done in the last 20, 30, 40 years on the cloud of microbes that live in and on people has focused on bacteria. And that's in part just sort of historical. That's what people tended to look at. But in the last few years, now that it's become easier and easier to do, in particular DNA sequencing, people have looked at a diversity of other organisms, including microbial eukaryotes and also fungi. And it turns out that we are covered in many fungal species or fungal lineages that were very poorly understood until the last few years. And this is an example. I'll give you two examples of this, but fungi, like many, I mentioned this before, there are some lineages of fungi that aren't very easy to grow in the laboratory or not possible to grow in the laboratory, and we've only really learned about them with these CSI, microbiology, DNA-based methods in the last few years. So I've mentioned this before, you can look at things in the microscope or try and grow them, but an alternative approach that can be very useful is to use DNA to characterize microbial diversity. And just like people have been surveying the human microbiome for bacteria, which is what I showed in most of the previous discussion, people have also started to look at fungi in the same places on the human body and try and associate, just like I mentioned with bacteria, trying to associate particular fungi with particular parts of the body and then with variation in health status within people. And there's definitely some indications in the last couple of years that people with different health statuses also have variation in what fungi you find on different parts of their body. And this is just a figure from a very recent paper that came out last year or 2 years ago where they did a survey using DNA to survey all the fungi in different parts of the human body, and in most cases, when they show all of this purple here, it's these particular sites, so different parts of the ear, the chest, the arm, the feet, are dominated by just one lineage within the Basidiomycota, this Malassezia lineage. So for some reason, there's this association between this particular lineage of fungi that we know very, very little about and many different parts of the human ecosystem. And so I think you'll see in the next few years a better understanding not just of the bacterial associations with humans, but also the fungal associations. This is a picture of one of them growing. There's just mostly relatively poorly characterized group of organisms. And I just thought I'd mention there's a more recent, even more extreme example of unknown fungi that came out, I think about 2 years ago or 3 years ago. People doing PCR surveys of different environments started to build evolutionary trees of fungi that were found in environmental samples. And they found a whole new branch, a major lineage of fungi that had never been described before, never characterized, nobody knew anything about it. And so if you look, this is a phylogenetic tree based upon, I think, largely ribosomal RNA genes, and you can see the Basidiomycota, the Ascomycota, the Zygomycota, the chytrids. This is another group that we haven't talked about. And then here's the Microsporidia down here, and then this whole lineage here, which they named the Cryptomycota, which were basically made up of organisms that seemed to be abundant in some environments, but nobody had worked with, nobody had grown in the laboratory, nobody had any real information about these. So this is, in the next version of the textbook, they're probably going to add another branch to the fungal lineage as people start to characterize this group that was completely unknown. Again, it's out there, people had seen it, but they hadn't been able to assign what they saw in the microscope to any particular group, and they were basically ignored because people thought, I think, that they were just yet another chytrid example. But they're very distinct evolutionarily, and as people study them, they appear to be biologically distinct. All right, so the use of fungi in food by humans is, there are many examples of this. In a way, you can view this as a mutualistic association, much like the ant example. People growing particular fungi are, in essence, providing good habitats for those fungi, food sources, water, protection from other environments, and the humans use them in some way. And I'm just going to give you a few examples of this. So I mentioned a few of these when we talked about the different lineages of fungi. So the Penicillium group, there's a group of Ascomycota that are found in high abundance in many environments. If you go out and pick up a soil sample and you try and grow the various fungi from there, or if you do a DNA survey, or if you look in the microscope, you frequently find representatives of this group in all around the planet. And also, they grow very well in human environments. So this is a particular species growing on an orange, and people have captured some of these and now use them extensively in food production. So camembert and brie cheeses come from a particular couple of species of Penicillium that are either purposefully added or come from the environment during the production of the cheese, and then convert some of the milk into the output of, particular desire to some people, which are these incredibly runny cheeses. Many of the blue cheeses come from another Penicillium species. It's got these blue conidia, and so this is the fungus growing, the Penicillium fungus is what's growing inside the cheese, producing the blue parts of the cheese that some people like, some people don't like. And then I mentioned this before too, but there are a lot of other species of fungi, not just the Penicillium group, that are used in a variety of food, fermentation, or production. There's an Aspergillus species that's used by many to make soy sauce. There's another Aspergillus species that's used in the brewing of sake. You have to be pretty careful, in particular with the Aspergillus species. I don't know of any Penicillium species that are particularly toxic, although there might be. But there are many Aspergillus species that are actual pathogens, and then other species that produce a variety of toxins. So you don't want to just randomly pick up an Aspergillus and make your home soy sauce brew without being careful about it. And then everybody's favorite, especially around Davis, is Saccharomyces cerevisiae. It's an Ascomycota, also known as brewer's yeast, and basically the main function that it carries out is to convert sugars into carbon dioxide and ethanol. If you're using it in bread, we like having the bubbles from the carbon dioxide, and the fermentation products are used, obviously, in things like beer and wine. And one thing is that it doesn't ferment to incredibly high levels of ethanol, because eventually when the ethanol gets to be particularly high levels, it inhibits the growth of the yeast. So you sort of has a self-selecting fermentation process by producing ethanol at the beginning, but then not when the ethanol levels get particularly high. And there's an incredible diversity of other examples of mushrooms and a variety of other fungi that are used by humans in food to eat either from the environment, or many of these are now farmed, although some people still haven't figured out how to grow some of these fungi very well in large-scale systems, so many are still harvested from the environment. And I think I mentioned this before, but there are some communities that actually have a diversity of lichens that they collect from the environment and even can sort of seed in the environment, and they eat those lichens. So it wasn't mentioned in the fungal farming story, but there's a component of the ant system and a component of many other fungal systems that's really interesting, which is that many species of fungi produce very interesting, highly complex, what are called secondary metabolites, which probably most of them are designed as either defensive chemicals or offensive chemicals in order to inhibit the biology of either other fungi or bacteria or sometimes animals. So it can be a defense of lichens, for example, to keep organisms from eating them. And they produce a lot of these compounds known as secondary metabolites. They're not necessarily essential for the normal day-to-day life of these fungi, but they play an important role in the ecology of many of them, and humans have taken advantage of many of these and isolated them and used them in a variety of ways, and I'm just going to give you one example of this, and it goes back to Penicillium species. So again, Ascomycota, ubiquitous in all sorts of environments, and this story is about this particular scientist 70 or so years ago, Alexander Fleming, and they were working in particular on bacteria. So Staphylococcus is a firmicute. They were doing studies of this Staphylococcus, and they would grow them in a variety of ways. This is what's called a lawn. So you take a culture, a liquid culture of Staphylococcus, and you pour it onto a little growth plate, and you spread it around, and then you get a growth of all, billions of Staphylococcus all throughout this plate. So it's a little layer of Staphylococcus on this plate that's called a lawn. And Fleming took some vacation time, as is, even today. And I don't think in the United States they're allowed to take vacation like this. But he took some time off and went away and left his plates out, just sitting out on his bench. And he came back, and there was some mold growing on the plate. That's not that rare. If you leave out a piece of food, mold will grow on it reasonably rapidly. But what he noticed was that around the region where the mold was growing, the bacteria were gone. The lawn of the bacteria was now missing. And he got very interested in this, eventually did a lot of work trying to figure out what this mold was, grew it in the lab. He could grow it, in isolation, look at it in a microscope, eventually typed it to being a Penicillium species. I think it was a relatively new species. And then they started to isolate this material that was produced by this Penicillium species. They called it originally mold juice. I don't think that was very popular as a treatment. But eventually they isolated the chemical, or collaborators of his isolated a chemical from this mold juice, and that was the first antibiotic, penicillin. And so what's going on is similar to the system that's basically going on in nature all the time. Fungi are trying to protect themselves from a variety of other organisms. They're also trying to kill competitors. Many of them secrete compounds, these secondary metabolites, that have detrimental effects on other organisms. And it turns out this strategy of what he did by accident has been responsible for the discovery of most of the antibiotics that we use even today. You grow up some bacteria, and then you take other organisms, either on purpose or an accident, and you grow them near the bacteria, and you see if they have any localized killing effect, suggesting that they're secreting some compound that might be killing those bacteria. And then you can grow up that organism and do different extractions. So what you do is you don't know what this organism is making that's killing the bacteria. You grow up a large batch of it, you crack open all the cells, and then you can separate out the different components of those cells into what are called fractions, different maybe pHs that precipitate out compounds, or different sizes of molecules, or different charges of the molecules. And then you take those fractions and you put them back onto the lawn of bacteria, and you can discover, eventually what chemical is being made by these organisms that are killing bacteria. And this has been how a vast majority of antibiotics have been discovered, by an experiment like this. Sorry, I don't know why I had to have a gross toenail slide on here. Penicillium species don't just kill other bacteria, so there are some species that if you do the same experiment with a lawn of mold or a lawn of other fungi, you can find that there are things that will grow on that lawn and kill in a little local environment the fungi. So it turns out that some of the antimicrobials that we use to kill fungi have also come from Penicillium species. And it appears that this genus just is very effective at making large amounts of compounds that are toxic to other organisms and then secreting them into their environment. If you remember, the way fungi work is they're these absorptive heterotrophs. They specialize in secreting compounds out of the cell that are to degrade other material, either living or dead material. So they're very adept at making compounds and pumping them out of the cell into their local environment, which we have taken advantage of to discover a variety of antimicrobial compounds. Again, there's a diversity of them that have come from fungi based upon experiments like this. Yeah, so this is just showing how it's really easy to do this type of experiment and find organisms that are producing some type of antibiotic. There are a lot of fungal species that are used in various traditional medicines. In many cases, people have then gone and looked at those particular fungi and isolated interesting secondary metabolites out of them. I don't know what fraction have been found, but the use of fungi for particular treatments in some cases has led to people to research those fungi and find a new secondary metabolite that has some interesting effects. So I just wanted to mention, this is now briefly, some other examples of where people are using fungi. So because fungi are really good at degrading dead or even living material, fungi are a key component of many processes where you want to convert biomass into some other product. So a simple example of this is composting, where really you're just trying to break up the biomass into smaller pieces and then individual constituents to then use that biomass for fertilizer or in some other way. And many people that do home composting, it's very effective to add into those systems various fungal lineages, fungal species, to increase the rate of this decomposition. On an industrial scale, there's a lot of examples of fungi being used to do the same sort of thing, but for example, it's called biopulping, to convert bark chips and other material into some other usable product. And now one of the big things is to try and convert biomass into fuels. There are people doing this with a variety of microorganisms. Some of them are algae, but there are also some fungi that are used in many of these processes. And again, because fungi are particularly adept at secreting a lot of enzymes, they can be very effective when used in a process where you want to degrade some material. You don't even necessarily have to isolate the enzymes from the organisms. You can just grow up a batch of the fungi and take the juice that comes out of them and apply it to your system. There are many places that now do basically this. It's referred to generally as industrial mycology. And I've mentioned this company before, but there are a couple of companies right in and around Davis that specialize in doing a variety of searches for new enzymes from diverse microorganisms. There's a company called Novozymes that is doing this. There's also a really interesting, I don't have a slide on this, but there's a collection of fungi that was made many years ago called the FAF collection that is now curated and kept at UC Davis. This individual went around and collected thousands of different isolates of yeasts and other fungi from around the world, and the people that work in that group, Curia Boundary Mills in particular, has tried to develop ways that people can rapidly screen these thousands of individual strains of fungi for various compounds that could be used in industry or agriculture. And so the last thing I want to mention here is just that we haven't really talked about fungi much in the environment, but clearly fungi play important roles in various ecosystems, either indirectly because of the role that they play in plants via mycorrhizae, but also directly because they actually, many of them have important roles. And one interesting thing, I didn't mention this much when we talked about extremophiles. There are some really interesting fungal extremophiles, in particular ones that can survive very low temperatures. There are some that grow at moderately high temperatures, but in particular, some of the really interesting ones grow at very low temperatures, and they have really interesting adaptations to allow them to survive in those conditions. And then the really important role that fungi play on the planet is in decomposition. So I've kept mentioning this, but basically the absorptive heterotrophy process that all fungi utilize allows them to participate in decomposition of organic matter on the planet, and they are the major decomposers of most of the organic matter in many ecosystems, and that allows for the recycling of nutrients, for the transfer of carbon down into the soil, for example, and they just play critical roles in the decomposition, in particular, of compounds like keratin and lignin and cellulose. And then the book has a little section that's kind of interesting about the evolutionary history, the role that fungi have played in particular periods of Earth history that may be a little more important than other organisms at that particular time. So one interesting thing is that at the Permian time, when the continents were coming together, there was a mass extinction event of many animals and some plants, but the fungi seemed perfectly happy and actually underwent a major diversification event at the same time. And I think I will leave it here. This is a picture of one individual Mycelium fungus that's growing in this forest. Just wait 1 second. That's the largest organism known on the planet. And I will stop. Yeah. I like the book. I told them I liked the book. I told them that it's my book. But I know that other people don't. So I said there is no other book that has a phylogeny approach. And therefore, it is, what we do here in 2C, it is optimal. But I know that some of the 2A people don't like it. They showed me their, adaptive software thing. Fine, nothing special.
I was just wondering if you were going to put the key up to the term.
Eventually, yeah, we will.
Eventually. Okay, when will that be?
I don't know.
Okay. Thanks.
Yeah.
Yeah, the 2A people don't. 2A is changing a lot.
To be not the John Roth model.
Yeah. It's
Back to the old model.
Discussions are changing a lot. They're